Welcome back to Imat Tutor. Today, we're diving into a really important part of Imat biology that often shows up in the exam: linkage and genetic translation. This topic ties together meiosis, inheritance patterns, and how genes are expressed at the molecular level. I'll walk you through the key ideas step by step, just like we're sitting together in a tutorial. I'll explain any tricky term as we go, and I'll point out exactly what you need to remember for the exam. Let's get started. Here's the big picture trap Imat sets. So listen to this as a logic story. Imat biology at the heart level is not about memorizing definitions. It's about integrating ideas that are usually taught separately. When recombination percentage is given, the examiner wants you to decide whether the genes are linked or not just calculate a number. When map distance is compared with observed recombination, the key issue is whether multiple crossovers are hiding the true distance. And when mutation is mentioned, Imat often checks whether you confuse amino acid count with amino acid identity, meaning the number of amino acids may stay the same while the sequence changes. The takeaway is simple: Imat tests decision making using numbers, not the number themselves. You don't need to worry about the jargons here. We will break down each and every important topic, and after the lecture, it will be crystal clear to you. Next, we define linked genes. When we say genes are linked, we're really talking about the physical reality, not a genetic rule. In this picture, the left blue chromosome has two genes. Both are denoted with the letter A. These two genes are linked genes. So we can say that if two genes lie on the same chromosomes, they are literally part of the same DNA molecule. So during meiosis, they are pulled, aligned, and separated together. Because Mendel's law of independent assortment assumes genes sit on the different chromosomes or far apart, that assumption quietly breaks down here. Let me remind you about independent assortment for a bit too. Independent assortment is basically the idea that one gene doesn't care where another gene goes during gamete formation. When meiosis happens, chromosomes line up in the equator, as we see in this diagram. One is blue chromosome, another one is red chromosome. If two genes are on different chromosomes, the way one chromosome goes into a sperm or egg has no effect on where the other chromosome goes. The red chromosome can stay on the right side, or it can also come to the left side. That means that the cell combination can look like this. There can be a cell where the red chromosome appears, or there can be another cell where the red chromosome will be on the left side. And same for the blue chromosome as well. The orientation will be random, and this is independent assortment. The gene for one trait is assorted independently of the gene for another trait. Because of this randomness, all possible gene combinations are produced in equal numbers. Capital A small a, capital B small b individual will make capital A capital B, capital A small a capital B, and small a small b gametes in equal proportions, and that we write as 25, 25, 25, and 25. That means all four of the gametes have 25% chance, or equal probabilities. And as we were talking about the linked gene, when there's phenomena of linked genes, we were saying that the assumption quietly breaks down here. The result is that parental gene combination appears far more often than expected in case of linked gene. The result is that parental gene combinations appear far more than expected, while new combinations appear less often. This happens not because the cell prefers certain genes, but because whole chromosomes move as single units unless something physically cuts and swaps pieces. That something is crossing over, and if no crossover occur between two gene loci, the genes stay together in the gamete. This is why gamete probability shift from 25, 25, 25, 25 pattern and become biased toward parental type. The core idea to log in is this: linkage is not a statistical trick. It's a consequence of genes being struck together on the same piece of DNA unless crossing over forcibly separates them. So we have covered that linked genes stay on the same chromosome, they are inherited together, and due to linked genes, gamete probability distribution is shifted, and the gamete probability we were saying was this: 25, 25, 25, 25. That means all the 4 gametes have equal probability of appearing. In previous slide, we were talking about crossing over, and we were saying that crossing over can cause recombination. This crossing over happens during meiosis, which is the cell division that makes sperm and egg. It takes place in prophase 1, the early stage when chromosomes first pair up, that we see in this diagram. Within prophase 1, there is a stage called pachyton, which simply means the phase where paired chromosomes are tightly attached along their length. Because they are pressed so closely together at this time, non-sister chromatids, meaning one chromatid from one chromosome and the matching chromatid from the other chromosome, can exchange pieces of DNA that we see in this specific event. And this exchange is called crossing over, and it physically swaps gene segments between the chromatids. As a result, new gene combinations are formed, and these are called recombinant, meaning chromosomes that now carry a mixed set of parental genes. The chance of crossing over depends on distance, because the further apart 2 genes are on chromosome, the more space lies between them, and the more likely a crossover will occur somewhere in that region. The main idea is this: crossing over is a DNA swap during early meiosis that creates new gene combinations, and it happens more often when genes are farther apart. Earlier I said that the chance of crossing over depends on distance. Now let's understand that in a bit more detail. Look at this image and think of each long horizontal line as chromosomes, and the X-shaped crossings as crossover events. Crossing over is a random DNA exchange that can happen at many positions along the chromosome when homologous chromosomes pair during meiosis. In the left part of the image, the 2 genes are far apart, that is, A and B. So the region between them is long. Because that region is long, there are many possible places where crossover can occur between the genes, which is why you see several crossing points. Each crossing point is another chance for the genes to be separated, so recombination becomes more likely. On the right side of the image, the 2 genes are very close together, as you can see, A and B. So the region between them is very short. A short region means very few possible crossover positions. So most crossover events miss that region entirely and the genes stay together. This is why genes that are far apart recombine more often, while genes that are close together usually remain linked. So the final takeaway is: distance doesn't cause crossing over, it increases the number of places where crossing over can happen between 2 genes. This statement highlights classic Imat trap. The statement is: crossing over always produces recombinant gametes, which is false. A crossover must occur between the loci of interest to generate recombinants for those genes. Let's understand what it means. As we have said already, crossing over means an exchange of DNA between homologous chromosomes. But that exchange only changes gene combination if it happens between the 2 genes you are tracking. A locus means the fixed position of a gene on a chromosome. If a crossover occurs outside the region between the loci of interest, the genes remain together in the same combination even though DNA exchange did occur. In that case, recombination has happened at the chromosome level, but it produces no recombinant gametes for those particular genes. That is why crossing over doesn't always lead to observable recombinants. The final takeaway is: a crossover creates recombinants only when it occurs between the specific gene loci being studied. To get a clear mental picture of the question that we are discussing, look at the straight chromosome line in the image. The letters capital A and capital B mark the position of 2 genes. That stretch of line between A and B is the only region that matters for recombination between these 2 genes. We already said crossing over means a random cut and swap of DNA, and that cut can happen anywhere along the chromosome. When we say the stretch of line between A and B is the only region that matters, we are being very literal. Genes A and B sit at two fixed positions on the chromosomes, like two pins stuck into a ribbon. Nothing about combination depends on the genes themselves. It depends on where the cut happens relative to those pins. Again, crossing over is a random cut and swap along the ribbon. If we consider this a whole ribbon, then the crossing over can be happening anywhere in the ribbon. The cut doesn't target A or B. It just happens somewhere. Now think of the chromosome divided into three regions. The region before A, the region between A and B, and the region after B. If the cut happens before A, both A and B are still on the same piece of ribbon, that is this ribbon. If the cut happens after B, here, again both genes stay together. In both cases, swapping pieces changes DNA elsewhere, but A and B move together, so nothing changes for these genes that are A and B. Only when the cut falls between A and B does something different happen. Now A lies on one side of cut and B lies on the other. Let's say the cut was here. So A lies on one side and B lies on another. And when the chromosome pieces are swapped with the homologous chromosome, A and B are no longer inherited as a pair. The single fact separating them onto different pieces is what creates recombination. This is why distance matters. If A and B are very close, the between region is tiny, so there are very few places where a cut could land and separate them. If A and B are far apart, the between region is long, giving many possible cut positions. More possible cut positions means a higher chance that at least one crossover will land in that critical region. So the region between A and B matters, not because it is special DNA, but because it is the only place where a cut can physically separate the two genes. So we can say a final sentence to seal the whole concept. Recombination between two genes depend only on whether a crossover happens in the DNA segment that lies between their position on the chromosomes. By them, I mean the genes that we're studying. Hope that easily explains the whole trap and statement. Look at this image and think of recombination frequency as something you measure from offspring, not something you see inside the cell. Recombination frequency means the percentage of offspring that show a new gene combination compared to the total number of offspring. And that is why it is calculated as recombinant offspring divided by total offspring multiplied by 100. Recombinant offspring simply means individuals whose gene combination is different from both parents. This number is observable because you can count offspring traits, but you cannot directly count DNA exchanges during meiosis. That is why recombination frequency is not a crossover count. Sometimes a crossover happens, but does not change the gene combination you are tracking. So it leaves no visible effect in the offspring. So we have covered the recombination frequency formula. Then we count recombination frequency with observable outcome. And recombination frequency is not a crossover count. Now think about map distance as a way of converting recombination frequency into a genetic distance. By definition, 1% recombination is called one map unit, also known as centimorgan, meaning a unit based on frequency, not physical DNA length. This works well over short distances because most crossovers that occur between nearby genes are detected as recombinant. Over longer distances, the situation changes because more than one crossover can happen between the same two genes. When that happens, some crossovers cancel each other's effects, making recombination appear lower than it really is. The takeaway? Map distance uses recombination frequency as a ruler. The diagrams just demonstrate that 1% recombination means one map unit. And the thing that we have said that it is a unit based on frequency, not physical DNA length. This image confirms that. And when we were talking about reliability, it is highly reliable on short distance. But as the length increases, we see impact of multiple crossovers, and as a result, the recombination frequency becomes less reliable. Now let's talk about an Imat trap. Imat may suggest that map distance equals physical distance in base pairs. We have already debunked that, but we will go into detail so that it becomes permanent in our memory, especially with this line being added to the trap. We have said that map distance is based on recombination frequency, and it meant how often two genes are separated by crossing over, not how many DNA base pairs physically lie between them. Base pairs simply mean the actual length of DNA. If we draw a DNA like this, these are the base pairs, letter by letter. So base pairs simply mean the actual length of DNA, letter by letter, along the chromosome. Imat may try to make you assume that more base pairs automatically mean more recombination, but that is not true. Recombination does not happen evenly along the chromosome. There are recombination hotspots, meaning regions where crossing over happens very often. Let's say this is a hotspot, and in this space there will be higher frequency of crossing over. There are some other regions where it happens very rarely. Let's say this is the region. Because of these uneven regions, two gene pairs with the same physical DNA length between them can show very different recombination frequency. And by this, we understand the last sentence. To decide whether genes are linked, we use recombination frequency as a guide. If recombination frequency is close to 50%, the genes behave as if they are unlinked, meaning they assort independently. Independent behavior means the genes separate randomly, just like genes on different chromosomes. If recombination frequency is less than 50%, the genes are linked, meaning they are close together on the same chromosome and usually inherited together. The smaller the recombination frequency, the closer the genes are to each other. And the recombination frequency ceiling is 50%. We're going to know why in the next segment. But before that, remember the takeaway message. Low recombination frequency means close, linked genes, while about 50% means unlinked behavior. The reason recombination frequency never goes above 50% is due to 50% ceiling mechanism. When genes are far apart, that you can see in this image, the distance is very high, multiple crossovers can occur between them. A double crossover means that two DNA exchanges happen in the same region, and the second exchange can restore the original parental arrangement of genes. That means the second event reverses the effect of the first event. As a result, recombinant types and parental types end up appearing in roughly equal numbers. This makes distant linked genes look unlinked, even though crossing over is happening frequently. So the takeaway is, multiple crossovers will hide recombination, creating a maximum observable limit of 50%. When we say that the recombination frequency reaches 50%, it starts corresponding with independent assortment. That might confuse some people. To clear the confusion, let's think of it from a different perspective. As the distance between two genes keep increasing, recombinant gametes and parental type gametes start appearing in equal numbers. At that point, half the offspring look recombinant and half look parental. That is the same pattern you get from independent assortment, where genes behave as if they are on different chromosomes. Because you can never get more recombinant types than parental types in this situation, the observable recombination frequency gets stuck at 50% and cannot increase further. And in this diagram, see how first crossover created variation, but the second crossover reverted the variation back to its original place. That's why double crossover can restore the parental configuration. This Imat trap exists because the word double makes people think more crossing over, so more recombinants. But biology doesn't work that way. A double crossover means two separate crossover events happening in the same chromosome region during one meiosis. Each crossover itself is an already uncommon event, So for two of them to happen in the same region is statistically much rare. Because probability multiplies, not adds, the chance of two crossovers happening together is far lower than the chance of just one crossover happening. That's why it is said that the most frequent recombinant class is not the double crossover. Double crossovers are the least frequent. Now we come to a more advanced concept, and that is 3-point mapping. When mapping three genes, your goal is to deduce their order on the chromosome. The rarest class of offspring usually comes from a double crossover. That's why it is written that double crossover is rare. By comparing double crossover offspring to the parental type, you can figure out which gene is in the middle. And we have already discussed that double crossover class is very rare. That's why it is written as least frequent. Now we will take a deep dive with this diagram to understand the concept of 3-point mapping in more detail. First of all, try to understand what the picture is showing. You have three genes on one chromosome, shown as letters along the chromosomes, and they are A, C, and B. We will also consider the right chromosome having A, C, and B. Capital A and small a doesn't matter in this case because they are just alleles, or they are just variations of the same genes. So the picture lists different outcomes. The first one is P, that means parental, and we don't see any crossover here. That's why it's written no CO. The second list is single crossover. The capital C is swapped with small c. Another possibility is again single crossover, but at this time small b is replaced by capital B, and that small b has come to this side. And finally we see double crossover, where two letters have changed. C has become small c. Now let's explain what's happening in the last row, that is denoted as DCO, that stands for double crossover. First, remember what DCO means. A double crossover means two crossover events happen in the same meiosis, one on each side of the middle gene. This is the middle gene. So crossing over will happen on the left side and the right side. Compare it with parental row at the top. The original arrangement was capital A, capital C, and small b on one chromosome, and small c and capital B on the homologous. This is the reference pattern. Nothing has been swapped. Next, look at the DCO row, which is labeled as least frequent. In this row, two swaps have happened, one between A and C, another between B and C. Because of the first crossover, the segment containing C swaps once. That's why you see that even after crossing over twice, only one gene has changed its location, and that is capital C has come to this place and small c has come to this place. And so what is the physical result? The outer genes capital A and B swapped twice, so they end up back in their original parental positions. The middle gene capital C is swapped once relative to the outer genes, so it ends up flipped. This is why in the double crossover row you see capital A still paired with B and small a still paired with capital B, just like the reference one. But capital C has changed from capital C to small c and vice versa. And this is the signature of double crossover. Everything looks parental except the middle gene. So from this whole explanation, one sentence takeaway would be: use the biggest class as parental, the smallest class as double crossover. This means this is the biggest class and this is the smallest class. And the gene that changes between them is the middle gene that we see here in this case. Now we are moving to the molecular side of our lecture. First of all, starting with codon basics. A codon means a group of 3 RNA bases read together during protein making. Because the genetic code is a triplet, the cell always reads RNA 3 bases at a time, never 2 or 4. These triplets are read in a fixed way, meaning once one triplet is read, the next triplet starts immediately after it. So codons do not overlap. The genetic code is called degenerate, meaning more than one triplet can point to the same amino acid. But it is not ambiguous because each single codon always means only one amino acid. The code is also described as universal, meaning most organisms use the same code with only rare exceptions. If we write AUG, this is a triplet and this is a codon. This is a start codon and it codes for amino acid methionine. As we were talking about degeneracy, degeneracy means that different codons can produce the same amino acid. This happens because there are more possible triplets than amino acid, so the system has built-in redundancy. There are in total 64 possible codons because there are 4 RNA bases and they are read in group of 3. But there are only 20 amino acids. Since 64 is much bigger than 20, the system has to reuse codon. The reuse is what we call degeneracy, meaning multiple codons keep the same amino acid. Here are clear examples. Take the amino acid glycine. It can be produced by these different codons such as GGU, GGC, GGA, and GG and G. Even though multiple codons can mean the same amino acid, the code is not ambiguous, which means one codon never codes for 2 different amino acids. This design protects the protein because some changes in the codon do not change the amino acid at all. So the takeaway is degeneracy adds safety by allowing different codons to produce the same amino acid without confusion. When we talk about translation, that means protein synthesis, this process must start and stop at the correct place, and this is controlled by start and stop codon. The start codon is AUG that we have already discussed, and this also codes for amino acid methionine, meaning protein synthesis always begins with methionine. Once the start codon is found, the ribosome begins reading the RNA in triplets. Stop codons are different because they do not code for any amino acid. Instead, they act as termination signals that tell the ribosome to stop protein synthesis. To get a more detailed understanding of this topic, I will suggest you to watch the translation video that I will put the link in the description. The reading frame explains how RNA is divided into codons. The reading frame is set by the start codon, meaning once AUG, as you can see in this image, is chosen, every base after it is grouped into sets of 3, such as ACG, GAU, CAG, and the list goes on. Because the ribosome reads 3 bases at a time, adding or removing just one base breaks this grouping that we see here. One extra base C is added. As a result, the previous amino acid sequence has been altered. We see that here it was prolamine, gliadin, etc. Right now it is histidine, isoleucine, arginine, etc. And the phenomenon that we have discussed, this is called frame shift, meaning all downstream codons are changed. As a result, the entire protein sequence after that point becomes different. So the takeaway is that the start codon fixes the reading frame and single base insertions or deletions disrupt everything downstream that we see in this image. Mutation changes the genetic message in different ways. First, talk about the substitution. Let's say this is a code. Randomly I'm writing AAG. When one base is replaced by another, such as U, this is a substitution. This can be silent, meaning the amino acid doesn't change because of degeneracy. Or it can be missense, meaning the amino acid changes. It can also be nonsense, meaning the codon becomes a stop codon and protein synthesis ends early. The frame shift mutation, we have already discussed that, but still some important caveats we don't want to miss. First, it is said that a frame shift mutation happens when bases are inserted or deleted in numbers not divisible by 3. This shifts the reading frame, changes all downstream codons, and commonly creates a premature stop codon. So the takeaway is substitutions may or may not affect protein, but frame shifts almost always disrupt it severely. Let me also warn you about this trap. It says that This trap exists because frame shifts are usually severe that we have said. So students assume they are always catastrophic. A frame shift means that reading frame shifts, so all codons alter the mutation change, which usually ruins the protein. But the keyword Imat tests is where the frame shift happens. If a frame shift occurs near the start of the gene, almost the entire protein sequence downstream is altered. So the protein usually loses function completely. If the frame shift happens near the end, only a small tail of the protein is changed. The main structure of the protein may already be built correctly. So function can be partly preserved or only mildly affected. This is rare but biologically possible, and that is why Imat words the trap carefully. All these ideas connect at a higher level. Meiosis creates genetic variation through crossing over and independent assortment, which reshuffle genes. Mapping uses recombination frequency or RF to infer gene order and distance. At the molecular level, these genetic changes matter because changes in DNA alter codon and codon changes alter protein. This is how chromosome behavior during meiosis links directly to protein structure and function. So the final takeaway is chromosome level changes affect codons and codon changes determine protein outcome. And with that, we complete our today's lecture and we have covered all the topics that we were supposed to. Remember, Imat loves to test disconnections between inheritance pattern, map distances, and molecular outcomes. Practice thinking it through step by step. If you want Imat biology notes that actually help you score, that are not bulky textbooks, not random summaries, but exam-focused, trap-oriented, high-yield PDFs, the link is in the description. Study smart, not hard. And if you found this video helpful, like, subscribe, drop a comment, and see you in the next video. Peace.